using imaging flow cytometry, which is also known as the AMNIS instrument. So the first thing to know about extracellular vesicles is that they are very small. So this slide is just showing you a schematic essentially to scale, where a HeLa cell would be about 20 to 40 microns in diameter, a lymphocyte much smaller at about 10 to 12, an RBC somewhere around 6 microns, and then EV are classified into 3 different groups. There are very large EVs. These are typically apoptotic bodies. They're about 1 to 2 microns. There are large EVs, which tend to be called microvesicles as well. Those are about 1 micron down to about half or 0.2 microns. And then there are small EVs, which is what this talk will focus on. Small EVs are also called exosomes, and they are about 50 microns to 200 nanometers. So EV are secreted by almost any cell type, and they can, they're secreted in any species, also in plants. They're present in all body fluids, and they can be found as well in tissue culture media in most circumstances. They have a lot of biological functions that are related to cellular communication. These include RNA shuttling and antigen presentation. EV are membrane-bound particles, and they may contain various proteins, RNA, DNA, and certainly they have lipid content with the membrane bound. There are a lot of potential clinical applications for EVs, which is one of the reasons for the interest in the field. In the field of cancer, they are being looked at for biomarkers of disease, and there's also some consideration that they might be able to deliver drugs to tumor by fusing with the membrane of a target cell. Proteins that are involved in the secretion process end up being on the membrane of the EVs. Those include the tetraspanins. The most frequently used ones are CD63, CD9, and CD81. You'll see some of the work that I'm going to show you today involves those. And there's also a lot of heterogeneity within the EV populations. They've been shown to contain different cellular components. And those components could be based on both active loading mechanisms and passive loading mechanisms. So the size of EVs is always a little bit surprising. I liked this graphic from this very nice review by Maria Panagopoulou. And what she's showing you here is a nanometer scale that runs all the way from molecular oxygen up to grains of sand. When we look at a leukocyte, then it would be about 10,000 nanometers, and bacterium at about 1,000, virus somewhere at around 100, EVs somewhere between 30 to 1,500 nanometers, which puts them not too much bigger than an antibody at about 15 nanometers. And you can see that some of the dyes that we use in flow cytometry are significantly smaller than that, but not as much so, not a negligible size as they would be with cells. So I thought I'd put in a slide to tell you about the universal characteristics that we can use to identify small EVs, because they are so small and we're trying to find them optically. So the EVs are always bound by lipid membrane. So that's one thing we can find. Their size is typically right around 100 nanometers for exosomes. And the function and the size are definitely related. Tetraspanin expression is pretty much universal across EVs, although they may not be the same tetraspanin molecules. The endosomal secretion pathway involves the formation of what are called TEMs, tetraspanin-enriched microdomains in the lipid membrane. And those tetraspanins will actually pinch off and form the membrane of the tetraspanins. So again, those are CD9, 63, and 81 across most sources. Other tetraspanins would include C82, CD37, 53, and the C-terminal domains are thought to be active, involved in active loading mechanisms. EV are heterogeneous in both transmembrane and their internal composition. So there are lots of cell surface markers that can be recruited to EVs. Often EVs are found to be MHC class II positive, so they're possibly able to present antigen within the immune system. Their marker composition is dependent on the tetraspanins. As I said, the variable C-terminal domain is going to influence that active loading. And there's also microRNAs that are known to be shuttled between cells with our EVs. There's lots of methods for EV analysis, and these include resistive pulse tracking and nanoparticle tracking, both of which get you what are considered to be the most accurate information about size and concentration of the particles. There's also cryo-EM, which works very well for accurate sizing and can give you some information about the heterogeneity of the particles. Live cell imaging is often used, although it's hard to segment EVs because they're so small that you can't really easily identify them because they're sort of at the 1 pixel range in most systems. A lot of protein methods are often used for EVs. Those would include like Westerns and IPs and ELISAs. That can tell you something about the population's protein expression, but not single particle protein expression. There's a lot of specialized systems that are in development, including surface plasmon resonance and others. Those are not widely available yet, and there aren't typically commercially available instruments to do that work. For a long time, people have been trying to study EVs using EV binding beads, so like beads that have a CD63 on them, anti-CD63 on them that can bind to the EVs. Those are very easy to set up and easy to detect with a flow cytometer. You cannot use them to count them because there's unknown numbers of exosomes that are actually bound to the beads. You can't see anything about the heterogeneity because it's random which exosomes will bind to the beads. And you're also probably sampling in an incomplete way since an anti-CD63 bead will not bind CD63 negative EV. Single particle EV flow cytometry is more complicated. However, the benefits are much bigger. They are useful to look at heterogeneity. However, it's challenging to separate small EVs down in that 100 nanometer range from the noise that you would see, and to also exclude single events that can be comprised of more than one EV. They're not thought to aggregate very much, but they can come through the interrogation point on the cytometer at the same time, and that means they would be recorded as one event. So that's what we call a coincidence event. A number of people have done sort of in-house hardware modifications to optimize flow cytometer performance for EV research. All of that is not standardized commercially. Then there's imaging flow cytometry, which is what I'm going to tell you about today. So the first thing I would tell you is that you need to use multiple methods. These EVs are hard to find, and none of the methods that are used are going to give you all the information that you want. So you can see here, this is a cryo-EM micrograph. This is information from a Zeta view instrument. So this is giving you size and counts. You can see that this particular EV was about 81 nanometers in this population, was the average size of these. And then this is what the imaging flow cytometry data looks like. And I'll show you a little bit more about that. So in order to publish this, you'll need to use a group of the methods that we just talked about. So why would you choose to do this using an AMNIS cytometer rather than conventional flow? A lot of this is because the diameter of the particles is so much less than the wavelength of light that we're using. If you look at the spectrum here, you can see that the EVs are well below the wavelength of visible light, which starts at like 350 nanometers. They're within the UV range, just slightly longer than an X-ray. And that makes them difficult to find because they have trouble disrupting the wave of the incident laser light. People have tried for a long time to detect them using scatter on flow cytometers. However, often the side scatter signal that you would use is essentially too big a wavelength to get a reliable signal. You end up with a lot of noise in your sample. Based on our research, we found that the limit for what we could see in standard flow cytometry was about 200 nanometer particle diameter. The AMNIS instrumentation works quite differently than a standard flow cytometer. It uses detection based on a CCD camera using a time delay integration in order to follow the particles as they move down the CCD camera array and get a longer runtime in front of the detector so that you can detect fluorescence quite sensitively. Our instrument is 5 laser, 12 channels. It has 2 cameras that are synced together using an internal bead. There are multiple objectives on our instrument. the AMNIS triggers off any of the channels. So it can trigger off the bright field, the side scatter, and the fluorescence. Anytime it sees a signal in that, it will record an event. The image that you see on the instrument, and you can see that hopefully on the screen here, is actually correlated with the dots in, that you see in the plots in the standard cytometry data. So that means that if you click on a dot, you can see the image for that particle. It was developed originally for subcellular localization, so the objective, the power of the magnification is not as high as you might think. We use a 60x objective. I can't remember the numerical aperture off the top of my head, but it's not a very high resolution system as far as the images go. It does provide volumetric data in terms of particles per mil, and it does that in every case because it's using non-pressurized fluidics. It's using a fluidics based on a, based on a volumetric pump. It was first published for EV application in 2014, so we're going back a few years here. The senior author on that was Joanne Lanigan, who was very helpful in this project getting us going with this. And I think we've made some improvements on this certainly since the 2014 publication. And there have been a lot of standards that the research community in EV research has published, largely through the International Society for Extracellular Vesicles and the International Society for Thrombosis and Hematopoiesis, as well as ISAC, the Cytometry International Society. So the first thing that you're going to need to do for detection of submicron particles on the AMNIS system or on any cytometer is to use a submicron sizing particle. And in this case, what I'm showing you are green fluorescent submicron polystyrene beads. These are commercially available. We've used two different suppliers, Spherotech and Thermo. I've included the catalog numbers here for anybody who wants them. In the top panel, what I'm showing you is the data off of our LSR Fortessa. So here you can see that we've got the green fluorescence here, the side scatter along the x-axis, and we can see the bead populations coming up in a nice tight distribution. Now there is about 100 micron bead in here as well, but I couldn't find it because it's buried here in the noise. When you pull these populations and you put them onto a plot for side scatter, you can get a standard curve because you know the size and you're measuring the side scatter, so you can infer what a side scatter measurement means in terms of particle diameter. On the AMNIS system, we're able to get a much more robust sizing of the particles. So you can see here that what we're looking at again is green fluorescence versus side scatter. And all of our particles are shown here, where we start at 100 nanometers, moving up to 2 microns of the largest size particle. This is what the images look like. So what we're looking at here is bright field here. And you can see that as you get into the smaller particles, you lose that bright field signal entirely. We can't see anything by about 200 nanometers for bright field. We can see green fluorescence. I don't know if you can see that little dot there. We can also see a side scatter signal for these, although it's very faint for what we would expect to detect for any kind of larger particle. It's barely a mathematical signal. We also, in this case, looked at side scatter off the violet laser as well, thinking that with the shorter 405 wavelength, we would get a better signal for side scatter than we would get with the 785. However, we did end up going with the IR for most experiments, and I'll show you more about that in just a second.
Karen, just hold on a second. I want to remind everyone to please, if you have questions as the process go along, put it in the chat on the group chat, please, in the public setting, and we can answer the questions. And if we, especially if you need clarification, then we can get to a natural point. Sorry about that, Karen.
No, not at all. Okay, so the submicron size standards that I was just telling you about, there's a few different things that are proposed to use for this. What we're using in this talk are green polystyrene beads. They're also sold as blank polystyrene beads. However, they're much harder to locate. And since you're using these as a reference particle, it's nicer to have them be fluorescent if you're using a fluorescent system. However, the problem with a polystyrene bead is that they very much have a refractive index mismatch compared with the EV. The EV are membrane-bound particles. They're pretty transparent to the laser, whereas polystyrene bead is quite opaque to the laser and scatters more light. So you're always going to overestimate the size of your EV on the side scatter scale when your standard curve is based on these. A lot of people have proposed using liposomes. They're much closer in refractive index, so you'd get a better, more accurate measurement of size. However, you can't find commercially available size fractionated liposomes, at least not that I have located. There was a proposal to use hollow silica beads. I haven't seen those commercialized, and we haven't looked at them. And then there have been a lot of other particles that have been proposed and have never come out onto the market. In experiments with small EVs, we're using these to estimate the size, and here's what our standard curve looks like here, where we have intensity of the side scatter signal here, and diameter of the particle here, and you can see that we have a nice linear range that runs all the way up from about 100 to 1 micron, 100 nanometers to 1 micron. When we compared the 785 dark field or side scatter versus the 405 side scatter, what we did see was a stronger signal off the 405. However, we got a lot more spread in that signal, and the linear range was not any longer, so we ended up deciding that the IR side scatter was a better way to go. The compression of the EV sample on the side scatter scale made it easier to distinguish the EVs from other contaminating things that were in the preparation.
We have a couple questions just out of curiosity. Some people are really interested in the baseline, discriminating these from background and comparing the CCD camera versus the PMT.
So it's hard to make a direct comparison other than that the CCD has a wider range, a much longer linear range than the PMT. And we definitely find differences in sensitivity, particularly toward the red end of the spectrum. I know that AMNIS claims that they have better sensitivity down into the green area of the spectrum. I don't know that I've seen that experimentally tested, and corporate literature is what it is, right? Were there any other questions?
We did have one on the quantification of the EV subpopulation with the image stream as compared to nano FCM, although I think you may have covered it somewhat.
Yeah, I think talking about nano FCM, it's a slightly modified version of a flow cytometer that's purpose built to be able to use for microparticle research. For us in the core, we haven't evaluated that very carefully. In a core lab, you really need to make a decision about instrumentation that can be used for a lot of different applications, whereas a lab that strictly did EV research maybe would want to make an expensive investment in a really single purpose instrument. Yep. So I can't tell you too much about the direct comparison there. Those nano FCM instruments do look like they're pretty good though.
There is one from Thorsten about is there a commercial viral standard for size, well, potentially using viral particles?
Yeah, I've seen that as well. I don't think that anybody has that sized in a way that within the field it's supported as a standard. I don't know how uniform viri are in size, so I really would have a hard time answering that question. Okay. At the time that we did this research, we had started with the polystyrene beads, and so we continued with the polystyrene beads. In my opinion, the Zeta view is the great way to get the size of the particle, and what I'm looking for here is to look at heterogeneity primarily. I think probably there's a few EV that I miss when we do this analysis. However, I think we get enough of them to be able to take a good sample down to somewhere under 100 nanometers, although it's hard, like we said, to give a direct cutoff because of the refractive index mismatch. All right, I'm going to go on from here.
All right.
Okay, so there's a lot of fluorescence labels that have been used in EV for fluorescence, and obviously one of the first of these is fluorescent proteins, and we have looked at fluorescent proteins with somewhat mixed results. I'll show you a little bit of that later. Initially, a lot of people started using annexin 5, which is a protein that binds to polystyrene, sorry, I'm going to mess that up, phosphatidylserine that is exposed on the outer leaflet of the membrane during the secretion process. There is some justification for that. However, annexin-5 is not a very strong binding protein. It's calcium-dependent binding, and I don't know that that would be the most consistent way to identify EVs. We mostly here are going to focus on lipophilic dyes. We know that EVs are membrane bound, and so I would say that that is the universally easiest way to consider that you are looking at an EV, given all the controls that I'm going to explain to you later. Other labs have also reported using fluorogenic dyes such as calcine and CFSE. Now these will diffuse through a plasma membrane. However, they are they require an active esterase enzyme in order to become fluorescent. And I'm not sure why you would assume that there would be active esterases in EVs. People have also tried nucleic acid binding dyes, including DAPI and Hirscht, and this is going to be dependent on the membrane permeance of the dye, as well as clearly the nucleic acid content of the particle, which is not 100% of them will be nucleic acid binding. You also will, if you saturate the nucleic acid, you still need a signal that's bright enough to be able to pull them back out of noise. So the lipophilic dyes were what we ended up going with. And the work that I'll show you today are cell mass dyes from Thermo. We felt like that formulation really didn't form a lot of particles, whereas like PKH, you could see a lot if you just looked at dye in PBS. Here's what the lipophilic dye labeling looked like the first time we ran it, more or less. This is data from the LSR Fortessa, and you can see that if we look at just buffer, we can see that we have noise on the cytometer. When we look at an EV prep, we start to see that there's events that are well above the noise. We don't really have a good idea of the size of these particles. However, there's not so much more than there were here. We're moving up from 0.78% to 3.9% in this larger gate. Once we've added the fluorescence, however, we can pull the EV away from the noise in two different directions, and that in and of itself is going to allow us to be able to more easily identify these. So you can see that depending on the titration of the dye, we're getting either less or more dye positive EVs in this prep. So a lot of people have asked when I've discussed this project about the instrument settings that you would use for the EV. Basically, on the AMNIS, you're going to want everything set to its most sensitive. So all lasers are on at max power. There is no sensitivity setting for the CCD cameras, only the power modulation on the lasers on this instrument. You need to have the 60x, your highest power objective in. The pixel size there is about equivalent to the size of the EV, so you're not going to have a lot of pixels being filled up. You want to run the lowest flow rate so that you get the tightest hydrodynamic focusing, and all your EVs line up as closely as possible and are as in focus as possible. You want a long runtime in front of the TDI CCD array, and that's going to give you the best sensitivity for fluorescence. There is a box intended for cells in the software that says remove speed beads, and I do not put that in because these are the speed beads, the unlabeled particles that help you synchronize the cameras and are QC particles on the instrument, and to me they help you find where your EV population is if there aren't very many of them. Also, you should know that this is going to generate large files. In our case, we would use a stopping gate that is something like this one shown in green, where we're getting most of the speed bead population, much of the noise here, and all of the EVs. We also run it without the extended depth of field. We tested this, that's another option in the AMNIS system, and we found that we really lost focus and weren't able to get a clear measurement of approximate size, which we're using as one of the criteria to determine what's an EV. So these are moving on to ovarian tumors and what the EVs look like in these tumors. And you can see that the images are not terribly impressive. We see nothing on bright field except for this particular event, which must be larger. And the cell mask orange dye here, you can see that we can see fluorescence on all of these particles and that it's proportional to the size, which is kind of what we would expect. The violet side scatter here, this was an early experiment where we were going with that. and the IR side scatter here, we've got signal in all of these. When we look at this as a plot, which is how we normally would look at it, this is the intensity of the lipophilic dye versus the side scatter, and you can see that you get a nice population that ends up being the lowest possible side scatter measurement, so below 100 nanometers approximately, and that is bright for this cell mask orange dye. These that we're going to talk about in this presentation are purified by ultracentrifugation, and there's a lot of things you could do with preparation of exosomes. I'm not an expert on that, but I would say that the prep is very much critical for getting good results from this kind of analysis. So when your images look as great as this one that I've blown up for you, how are you going to know that you're looking at EVs? Unfortunately, the answer to that is the one nobody wants to hear, lots and lots of controls. You have to run a vehicle-only control. You have to run the EV prep with no dye in it so that you can establish that that's not some kind of inherent fluorescence or autofluorescence that you're seeing. You need to run the dye only so that you can tell that there's no aggregates that the dye are forming, because it is a lipophilic dye. It's also going to be hydrophobic, and it will form little balls potentially in the in your PBS. You need to use any fluorescence reagent that you're using. So if you're using an antibody to stain these, you need to make that only in the buffer without EVs. You're also going to need to run a whole serial dilution series of your EV preparation, and that's to establish that you're looking at single EVs. I'll show you more about that in a minute. You need a detergent control, because one of the things we know about a membrane is that you can lyse it with a detergent. So when you add a detergent to these samples, all of your EVs should disappear. And you also need to carefully titrate the reagents that you're going to use for this kind of an analysis. So I was telling you a moment ago about serial dilutions. The reason that we're doing these is because we can't easily tell if we're looking at single particles or two that happen to come through at the same time and were counted as one event. What we expect to see is that if we do a series of tenfold dilutions, we expect that it will dilute out in that the EV counts will dilute out in objects per mil according to the dilution series that you've run. If you get a nonlinear portion of this up at the top, you know you are too concentrated and need to dilute that prep out to make any measurements. We also look at the aspect ratio. So this is based on the lipophilic dye signal, and we're looking at the XY coordinates and pulling anything that is below 0.8, so that's not essentially round. Okay, the detergent control. This is important, like I said, because anything that lyses is likely to be an EV. Anything that does not lyse with detergent is unlikely to be a membrane-bound EV particle. All of your preps are going to have non-EV components that'll come through. Those will include lipoproteins, extracellular RNAs, possibly dye aggregates. If you use an MP40 or Triton X100, or what most people use, you can lyse those membrane-bound EVs, but you wouldn't be able to lyse a lot of your other aggregates of dyes with that. So we work on a number of conditions, and for most preps, we prefer a 0.5% MP40 mixed in equal part with the EV prep at the highest concentration and left at room temperature for 1 hour. Don't vortex that, because it's going to form bubbles, and bubbles are detectable particles. So you've run all your negative controls, and now you want to see what this data looks like. This is what a clean data set would look like. You've got all of your controls, your PBS by itself, your unlabeled EVs, your detergent control here, your 1 to 10 EV prep here, and then your cell mask alone with detergent and your cell mask alone without detergent. And you can see that when we plot this on a log scale, we get essentially nothing in any of the samples other than our labeled EV here, which have 50 million events or objects per mil. Other positive controls that you could think about would be to measure tetraspanins and other known components of EVs to confirm that that's what you're looking at. And of course, to go back and use several methods to confirm that these are EVs and that you're measuring single particles. So your counts here may not match exactly with what you see on a Zeta view, but they should be within a reasonable ballpark. So I told you we tried this with fluorescent proteins, and we had mixed results. So you need a lot of the fluorescent proteins in the particle, which does not seem to happen with a passive loading situation, but does happen potentially with active loading. So with a fusion protein of GFP and CD63, one of the tetraspannins I told you about, we could see green fluorescent EV from ovarian cancer derived EV prep. However, we looked at RFP and they could find this by Western in both the EV preps and the cells. However, there's not enough particle or molecules here in the EV population to be able to use this as a marker for EVs. This is largely the reason that we ended up going with the lipophilic dyes, is that it doesn't tend to be influenced so much by what's in the particles. So when you do antibody staining, you really want to consider what you're doing as far as the size of the dye. You can see that dyes do vary quite a lot from an organic dye, which is quite small, to a fluorescence protein, which is quite a lot larger. Antibodies even are big, and then a dye like PE or APC or PERCP is quite a large protein dye. Some of the organic dyes, like a FITSI or the Alexa series of dyes, are small molecules that are in proportion to the EVs, not going to cause a steric hindrance issue with staining. You'll be able to saturate the staining on that particle. So here I'm showing you antibody titration. This is, we're using CD9, which is one of the tetraspannins, and we've got our EVs at a couple of different dilutions here, and our antibody at a couple of different dilutions. And what we can see here is that in the cell mass green, if we dilute out the EVs, we can see that the concentration goes down. Same is true if we, same is true here for the green bars. And then if we use a high concentration of antibody, we don't get as strong a depletion of signal. However, if we use a low concentration of antibody, we start to flatten out the signal and probably are not saturating. So there's a lot of negative controls that we've seen with this, and I'm going to show you a different view of this data here. So this is, again, the same data set where we're getting CD9 positive exosomes that are at that are consistent in terms of the percentage that they make up out of the total EV population. And when we look at the intensity, with the higher concentration, we were able to get an intensity that was proportional to the EV dilution factor. Whereas with the lower concentration, it was flat all the way across and probably not saturating. So this 1 to 40 concentration, or 1 to 5 concentration that I show you here is what we ended up working with for the CD9 antibody. So another point to make is that when you've stained them, when you've stained EVs with both the fluorescent lipophilic dye and the antibodies, we want to make sure that the percent positive stays pretty consistent as you dilute them out, and that the objects per mil of the antibody positives will also dilute out. That confirms that you're seeing a percentage that is consistent depending, or with a variable number of EVs, and that you're not seeing coincidence events of one positive and one negative. So with CD9, here's what this data looks like. We're staining with CD9 B450, and you can see that we have our NP40 control here, looking at the cell mass green in this case versus side scatter. And with the NP40, we don't see anything in our gate. When we look at the cell mass green, we do see that. And when we take this gate and look at a histogram of the CD9, we can see that a good proportion, somewhere around 40–45% of the green particles are staining for CD9, a good confirmation that these are probably EVs. I'm showing you the images here, and you can see that we can see the signal for the fluorescent signal for the CD9 in the images when we're running at max power. So it's a mathematical signal, it's also a visible signal in the images. So there is heterogeneity between different EV populations. In this particular cell line, you don't see very much in the way of CD9. So this was a different cancer cell line. The last one I showed you was ovarian cancer, OVCAR8. This one didn't happen to have very much CD9 in there, whereas it did express a lot of CD81. I'm not showing you that data. So just one of the tetraspandins is probably not enough if you're trying to use them to confirm that those particles are what you think they are. So the linearity of the dilution series for the overall particles should be consistent. The dilution series should also be consistent for the stained particles. And the min-max should follow along with what you would expect, also diluting out with the dilution series. So interestingly, we were able to enrich specific EVs based on an immunoprecipitation. So this is a study that was done by a great postdoc who has recently left MD Anderson, or a great student who has recently left MD Anderson. And he was developing an IP method for these. So you can see that in this case, we are getting, when we do a control IP, we get many fewer cell mass positive EV particles. When he did the IP using his particular protein of interest, he was able to get a lot of particles to come down with that, and they expressed that protein of interest. With the NP40, the vast majority of these go away. And so that's a pretty significant enrichment from about 0.2% up to probably 3 or 4%. We were also able to establish that we could see these particles in biofluids. It's a much more complex mixture, which is why you've got many more populations and particles here in intermediate size ranges. And these are probably lipoproteins and other kind of biochemical components. This particular biofluid was ascites from an ovarian cancer patient, and it's known to be a pretty messy fluid. However, it fulfilled all of our other constrictions to be able to call these EVs. Their PBS control was negative, the NP40 control was negative. They did lyse. So maybe many of these are actually larger EV populations. This is the publication that I've been explaining, which I've been talking about today. This recently came out in November of last year from Dr. Emil Sud's lab, and you can see some of the figures here. You should check it out in Science Advances, just to plug since I'm using the data from the paper. And the conclusions here are that we can see the EVs exosomes by AMNIS imaging flow cytometry. The prep of the EVs is critical. I have shown you very nice looking EV preps that are clean and easy to gate, easy to tell what an EV should be. I've also shown you that ascites prep, which was quite a lot messier. I've seen many preps that were not easy or possible to analyze in a way that I was comfortable with. You want to carefully establish all the concentrations, the dye concentration for the lipophilic dye, and you should be doing that with the vehicle control and with the dilution series of the dye itself, and cross-diluting with the EVs so that you can see that you've got consistent intensity. You want to use sizing beads to determine your maximum size cutoff. As I said, the refractive index mismatch means that you're going to figure that you can't see any measurement for side scatter below about 100 nanometers. Those serial dilutions are critical to provide confirmation that you're looking at particles that dilute out, not aggregates of particles or coincidence events. And they should correlate between the EV percentage. If you're staining for a marker, that should also dilute out consistently. You want to carefully titer all your antibodies and any functional probes that you happen to be using, including DNA dyes or RNA dyes, that kind of thing. You want to look at the free dye and detergent controls, and you need to look at those in every run. I will say for the sizing beads on our instrument, we did find that those were extremely consistent run to run, and so in most cases we were able to run just the 200 nanometer bead to say, okay, the instrument looks like it's right on track with where it has been. We were doing these 3 and 4 times a week for a while. There's lots and lots of resources out there that you should take a look at if you're considering this kind of work. So as I said, the ISEV has been putting out a lot of information about best practices since there was a lot of Wild West kind of research that was happening in this field. So there are standards for the information that you should report for flow cytometry in EVs, a position statement here from the ISEV leadership about how they should be named and what the functions and claims that you should make in a paper would be. a comparison of some isolation methods that you might use for an EV prep, and also another one from ISTH, which publishes circulation research. That one was also a nice paper to look at. Compliance with reporting standards is a little bit variable. So if your paper is reviewed by journals that are run by ISEV or ISAC or ISTH who develop these guidelines, you are likely to have reported most or all of the things that you should be reporting in these samples. If you are publishing in other manuscripts, you may or may not be, but reviewers will always become more critical of research. So by the time you finish your project, maybe you can't publish it in Science. The next steps that we're looking to take are to look at intravesicular protein heterogeneity. Now that's non-trivial because staining proteins within a tiny, tiny particle is going to be pretty complicated, and we've had some good and some bad luck with that, which I'm not going to tell you about today. We're also trying to look at microRNA heterogeneity within the EV populations. There's a few ways that you could think about doing that. And we're hoping that we can enrich the EV populations by sorting. We were particularly interested in doing that because the populations of interest in that Science Advances paper that I showed you were about 0.1 to 0.5% of the total EV population. So a little bit hard to study. They were hoping to get those isolated so they could do further experiments with them to look at their function. So my acknowledgement slide, I really should thank the SUD lab. They're the people who actually drove the project in our lab. And so Emil Sud, the PI, and a very talented MD PhD student named Alejandro Vilar Prados, sorry about my Spanish, who has recently returned to finish his MD at the University of Puerto Rico. And Akira Yokoi, who is a postdoc in the lab and has really finished off that paper in a nice way and is continuing to do some interesting work with that. I got a lot of great advice on how to do this from Joanne Lanigan at the University of Virginia. She's the one that published the first publication with this, and from John Nolan at the Scintillon Institute, who's also an authority in this field. And obviously I need to thank everybody in my lab, including in particular Vina Papana, who has been in our group for a number of years now and who is our most experienced AMNIS user and did a lot of the hard work in getting some of this data analyzed and collected. And I had some grant support for this based on the NCI CCSG through MD Anderson. So that would be my talk today. I can take any questions.
Thank you so much, Karen. That was absolutely wonderful. So we have had some questions. I did have one quick question. I noticed that you're thinking about going forward sorting, and a lot of times with sorting, you end up diluting things. Are you thinking potentially even going so far as a magnetic bead step where maybe you delink the antibody so you can maybe concentrate the EVs a little better?
Yeah, so I mean, our initial thoughts on that are that we may go back to the ultracentrifugation to actually increase the concentration, but yeah, they do come off the sorter really dilute. It's a little bit hard to be sure that you're getting what you think you're getting off the sorter, but we have a lot of plans for what we do after that that makes it easier to determine the answer to that. We have done some post-sort analysis with the diluted sample, and it's actually better than I expected, so we'll see.
All right, we do have some questions from the audience. We do have someone asking about whether you're washing the EVs after labeling or if it's a wash-free.
So they do get washed after the cell mask application. The lipophilic dye does require a wash step. We just weren't able to find a titer that was consistent when we did not actually run through an ultracentrifuge step. So he would add the lipophilic dye prior to the last ultracentrifuge. For the antibodies, we were not able to actually wash the antibodies off, and that was one other reason that it was so important to get the titer right, because you want to be saturating, but you don't want to have antibody aggregates that you're detecting on the instrument.
We have one question about that. That's a process question of used from David, when you isolate the ovarian cancer EVs, did you use exosome-free FBS?
Yes, I think these are actually serum-free entirely.
Serum-free entirely, okay.
Yes.
We do have someone who's interested about how much for a system, and I can fend that one. That's somewhere between 150 to 500,000, depending on what you get. Unless you want to make a comment.
For this, I think you're closer to the $500,000 end of that.
Yeah, of course. Oh yes, definitely. Always a good reason why you should partner up with a core if you're at least starting off your experiments. We do have a question about aspect ratio. Will the dye affect the scatter, the dark field, and the aspect ratio? And does that need to be corrected for?
So no, the dye does not impact the aspect ratio for the scatter, but we actually use the impact ratio, or we're actually using the ratio for the cell mask dye, the lipophilic dye, rather than the aspect ratio for the side scatter. Mm-hmm. The reason being that we think that is a more direct signal. It's a little bit more in focus. It's a stronger signal, so it's a little bit easier to see where it is. We've never, in a good EV prep, we never cut out very much based on that aspect ratio gate. In a messier EV file, you might end up cutting out 80% of your events, but then I think you're looking at going back to how can I improve this prep?
We have a question on additional labeling additional proteins on exosomes. Maybe is there a point where you can't get any more antibodies on, or?
So I would imagine that there is that point. I don't know for sure if you would reach the limits of the AMNIS instrument first, or if you would reach the limits of the antibody first. I will tell you that we have done, in unpublished experiments, several antibodies and gotten decent staining intensity and histograms that were believable. And I think at some point you're probably splitting your samples so that you can't say, well, it's positive for this protein, this protein, this protein, and that protein. But you can say that I have some that are positive for, I think you're running multiple panels maybe. We've done probably 4 or 5.
Yeah. Now you talked a little bit about it, but they just want to clarify which the threshold of expression detected with this method. Do you know what kind of the lower limit is? Like perhaps using one of the brighter dyes, how many surface proteins?
Oh gosh. So we haven't done any kind of analysis of the number of protein molecules in the particles. In a lot of ways that it's out of the range of the normal MESF beads that you would use to do that. So you'd be sort of extrapolating a standard curve that you got from MESF beads with the known dye concentration, be extrapolating that way down in order to get to where these are. I think that there isn't really a known dye concentration standard that should be similar to this. Yes, so no, yes. I don't have a good idea for that.
That's tough. That's a tough one. I do have someone who wants to clarify, if you could clarify aspect ratio, whether it's a measure of roundness or something.
It is a measure of roundness, although in this case, we're using it less of a measure of roundness than a measure of the fact that there's one particle per frame. We're not segmenting these images, right? So you can imagine that if you had 2 EV in one image, that your aspect ratio for the fluorescence would be way off from 1.
Good point.
So a coincidence event where they're not actually physically bound, but they are captured in the same image is what I'm talking about. We want to make sure we exclude those.
All right, we have, let's see, there's just a couple more questions here. I think we have time for at least for one. There's someone asking if it's difficult to actually, or if it's possible, I would even go far, to perm the EVs to study anything intravescular.
I don't have a good answer to that now, but I think we could revisit this some time from now, and maybe. have a good answer. So I wouldn't say it's impossible, but it's non-trivial.
Yeah. Yes, it seems like it. In any type of permeabilization, you'd potentially lose your fluorescence.
It would be a pretty fine line to be able to perm them, and then you're thinking about fixative steps and washing, and yeah, it's not easy.
All right. Well, that's.
And then there's also, how do you standardize that? How do you know that you have that target in your EV to begin with?
Well, we've got a lot of. Sorry, I didn't get to everyone's questions, but thank you all for coming, and thank you so much, Karen, for presenting this wonderful information. It's really truly nice. It's such a great technical talk, and it's such a great introduction to the powers of imaging cytometry.
Okay, and if there's any questions that somebody would like information on, I'm pretty easy to find. You can either email me directly, and you'll find my email on the MD Anderson website, or you can look for me at the FlowText website, and you can find me that way too. So please do reach out if you have further questions, or you have something that you're just dying to give a try to, okay?
All right.
Thank you everybody for coming during the pandemic.
Thank you.